Welcome back to the biology classes at Class It. Today we are continuing with the topic AIDS. Today's session we will be dealing with symptoms of AIDS, treatment, prevention of AIDS, as well as some questions which may persist in your mind regarding the topic AIDS. Let us first proceed to the symptoms. The common symptoms of AIDS are recurrent types of or bouts of fever, diarrhea, then sometimes chills, rapid weight loss probably due to frequent diarrhea. Then there is a decrease in the immune cell count. We have already studied the AIDS virus affects CD4 plus cells, that means T helper cells, macrophages, monocytes and dendrocytes. So the number of these cells in your blood will start decreasing as the stage of the HIV infection proceeds. And by stage 3, you will have a very low cell count of CD4 plus cells. You can see the numbers here. Below 200 cells per millimeter cube of blood will indicate that that person has proceeded to stage 3 of HIV infection, which is actually called the AIDS. Until then, it is referred to as human immunodeficiency viral infection. So, by the stage 3, the number of T cells and macrophages particularly will decrease in cell count when compared to that of a normal person. And this in turn leads to a decrease in the immune functions. That means you can say leads to an immunodeficiency. As a result, the person will fall prey to many opportunistic infections. Now, what is meant by an opportunistic infection? You may have a doubt. An opportunistic infection simply means the infections which occur with more frequency or more severity in people who have human immunodeficiency viral infection or AIDS. In such people, some diseases which may affect a normal person and go unnoticed will proceed to a more severe version. For example, some people will have fungal infection of the tongue. Our body's immune system will effectively fight it off. That is called candidiasis. But in the case of a person with AIDS, this will proceed to the mucosal layers in the mouth, esophagus, trachea, lungs and so on, and progresses to pneumonia, may even lead to death, which starts off in a normal person as a simple infection which we can easily get rid of. In the case of a person with AIDS, that particular infection proceeds to a severe form. Similarly, they may also fall prey to infections from mycobacterium, for example tuberculosis, which again leads to death. Then fungal infections like candidiasis, you can see the names, cryptococcosis, histoplasmosis and so on. You need not remember the names, you just remember that they are prone to fungal infections. Then they are also prone to infections from toxoplasma, which is a parasitic protozoan. Toxoplasmic infection leads to encephalitis. Encephalitis is inflammation of the brain tissue. So all these things can eventually progress to conditions like pneumonia. Usually bacterial, viral or fungal infections will proceed to pneumonia, leading to death of the individual in the final stage of AIDS. You can see two pictures which shows you the progression of fungal diseases in the case of AIDS patients. See how severe it has become. This might have started off as a simple infection in a normal person which you can easily get rid of. But in the case of a person with AIDS, it proceeds to a very severe version. Now going to the treatment for AIDS. The treatment for AIDS is always changing. Over many years, many therapies have been used in the case of AIDS patients. The commonly used treatment for AIDS patients is generally known as antiretroviral therapy or ART, abbreviation for antiretroviral therapies, ART. It can be different types of therapies, and common type of therapy is to inhibit the reverse transcriptase enzyme of the virus. So those class of drugs which inhibit or which stop the function of reverse transcriptase are known as nucleotide reverse transcriptase inhibitors. You can see a couple of examples like abacavir, tenofovir, etc. Also, some other drugs may cleave the protein components of the virus when it infects the cell. You know that as viral infection proceeds, it produces viral proteins. So these proteins can be cut off by using proteases like entrinavir, indinavir and so on. And there is also therapy which is known as HAART, H-A-A-R-T, which stands for Highly Active Antiretroviral Therapy, which is usually used in case of people with AIDS and cancer. Some people will fall prey to cancer while they have AIDS. Now going to the preventive measures for AIDS. Always we know prevention is better than cure. And in the case of AIDS, mostly it is ignorance which leads to the transmission of the disease. So don't die of ignorance. That is the key sentence in the case of AIDS. In the case of preventive measures, people are made aware of these measures by certain organizations which are functioning exclusively for AIDS awareness. The name of such organizations are, one is NACO, which stands for National AIDS Control Organization, which is in India. Then several non-governmental organizations. You can see the list at the end of the session. You just have to remember there are several NGOs catering to the AIDS patients and making others aware of the transmission of the disease. And of course, the World Health Organization plays a very significant role in spreading awareness regarding the transmission of AIDS. You can see the steps to prevent AIDS, which have been put forward by several organizations and the medical people. The NACO and NGOs are educating people about AIDS. WHO has also started a lot of programs to prevent the spread of AIDS. You can see the measures which are taken by them. They have taken measures to make the blood from blood banks free from HIV. That is, we know that when a person is giving blood, that means when a donor is giving blood, he has to undergo several tests which determines whether he has a particular disease or not. In the case of AIDS, the screening test is usually to screen for antibodies. But the screening for antibodies may be negative in the initial stages of the disease. So advanced techniques like real-time PCRs have to be employed to see whether the person is actually carrying the virus even in the dormant stage. So tests like that are now advocated to make the blood safe in the blood banks. That means advanced tests are being recommended by WHO. Secondly, they have also made disposable needles and syringes, and they have made it mandatory that all the public and private sector institutions which are using these blood samples should use such kind of disposable needles and syringes, because we know the blood which is dried up or which is present in the syringes and infected needles can transmit the disease from one person to another. Advocating safe sex by promoting the use of condoms, especially among sex workers. Then controlling drug abuse. Then promoting regular checkups in HIV susceptible population. This includes the migrant population, those people who are involved in sex trade. So HIV susceptible population is identified and they are recommended to undergo tests to determine whether they are positive or not. Then finally, social awareness is very important. We have many myths and misconceptions regarding AIDS which has to be overcome. Now, a collective working of the medical fraternity along with the social workers are needed to tackle this malady. So we have to work very prudently to help in eradicating the disease to a zero level in the community. Now coming to certain common questions. The medical science has improved dramatically over the years, but still we have been unable to find functional cure for AIDS. When I say functional cure, it means a different thing. In the case of AIDS, there are two types of cures. One cure is known as functional cure. The second cure is known as sterilizing cure. Functional cure is where you are eliminating the disease symptoms, but the virus might remain dormant in your system or your genome. You are unable to eliminate the virus as a whole, but you are able to sustain the person who is suffering from AIDS. That kind of cure is known as functional cure. We have been successful to a certain extent in maintaining the functional cure, but sterilizing cure is a different thing. There you are trying to eliminate the virus as a whole. That means the virus should not be present in the individual. The infected individual should be 100% free from the virus. A sterilizing cure is yet to be determined. So medical science is trying ways to find a sterilizing cure for AIDS. Now the reason why we have this problem, okay, the AIDS virus is very brainy, you can say. The AIDS virus has two problems. We find it difficult to tackle those two problems. One, it remains hidden in our genome. In the dormant stage, it may not even produce any symptoms, and the person might not even know that he is or she is infected with the virus. Secondly, while the virus replicates, we have already studied the replication cycle. So in the replication cycle, we know that the viral genome enters the body of the host, and the RNA genome of the virus is converted to DNA, double-stranded DNA, with the help of the enzyme reverse transcriptase. Now, during the conversion from RNA to DNA, the enzyme reverse transcriptase is adding nucleotides. But there is a problem. This reverse transcriptase does not have proofreading. That means it is unable to determine whether it has added the correct nucleotides in order. In our body, when we synthesize a DNA strand, we do it with the help of the enzyme DNA polymerase. It has an extensive proofreading mechanism. But in the case of RNA being converted to DNA by reverse transcriptase, the enzyme reverse transcriptase does not have this proofreading. So the RNA from the virus, though it is converted to DNA, we can never be sure it is the same sequence which is being converted, or the complementary of that RNA is being synthesized. We are not really sure. So that is one problem. That is RNA being converted to DNA in the processor. There is a problem with the reverse transcriptase, that it is bound to make mistakes. That is, its error rate is very high. Similarly, the DNA of the virus is integrated into the genome, and from there it is transcribed again to RNA. Many RNA copies will be made to produce new viruses. Conversion from DNA to RNA is done by the host itself. The host RNA polymerase converts the DNA into RNA. But our RNA polymerase is also prone to errors. That means it has a very low fidelity. Fidelity means we cannot say the RNA polymerase will transcribe the DNA exactly. The error rate is also high. So there are two reasons in the genome itself which calls for a high mutation rate of the virus. It is also seen in advanced studies. For example, in Lancet, it was published that there is an enzyme in our body which attacks the viruses. When our body is finding a viral genome inside it, it will try to somehow modify it to get rid of it. It is actually a part of our defense mechanism. And this enzyme will cause mutations in the viral RNA with the hope that it can get rid of that viral RNA. But in the case of AIDS virus, it makes use of this mechanism. It makes use of our defense mechanism to create more and more mutations in its own genome so that it can survive in the host cell. You can say, as a total effect, the mutation rate of RNA in the HIV is very high. As a result, whenever we try to invent a medicine and we put it to clinical trials, this may take several years. By the time we are out with the drug, the RNA of the virus will have mutated so that it can escape all these responses. Another common question is how long a person might survive with AIDS. Average estimate says that if a person is infected with AIDS, say by 20 years of age, that person will continue healthy life with AIDS for about 35 more years. That is the average calculation given by WHO. That is, if the person is on suitable antiretroviral therapy. In the case of coronavirus, we read in papers that the time span during which the virus stays outside the body of an individual is, initially they said it was 8 hours, later they said it would be around 20 hours. In the case of AIDS virus also, you may have such doubts. At different conditions, the virus remains in the atmosphere at different lifespans. For example, in the case of cold temperatures, HIV will thrive for more hours. But as the temperature rises, the HIV will not persist in that higher temperature. Then at pH 7.1, which is the optimum pH for the virus, it can live longer. In the case of blood in syringes, it can stay up to 6 hours in a dried blood sample. But if the syringe contains fresh blood and if it is not exposed to air, they say it can last till 20 hours. The virus will be active for 20 hours in a syringe containing blood which is not exposed to air outside. So different lifespans are there. Up 11 days is the average lifespan given in research papers for virus to survive in air. Which cells do AIDS virus survive? That is another question. This you are already familiar. AIDS virus will survive in CD4 plus cells. That means those cells which have a glycoprotein called cluster of differentiation 4 along with certain co-receptors which are called CCR5 and CXCR4. The cells in the human body which have these receptors are T helper cells, macrophages. Remember these two as the main cells. Also monocytes and dendritic cells. Another question which can come to your mind is, why does saliva, sweat, urine and fecal matter not spread AIDS? The reason for this is not yet clear, but they say that at the pH of these body samples or in this body fluids and all, the AIDS virus will get disrupted. There is a hypotonic disruption of the AIDS virus. The cell will burst due to lysozyme and some enzymes present in these fluids. As a result, the AIDS virus will not be able to survive in these fluids. Another question is the number of AIDS patients in India. Now, AIDS is an epidemic. I hope you have found out the difference between pandemic and epidemic by now. AIDS is the third largest epidemic in the world, and India has 0.2% of people being affected with AIDS. 0.2 is actually a very small amount, but considering the Indian population, this will amount to around 2.1 million people infected with AIDS as per census in 2019. Another very interesting question, has any person been cured of AIDS? Yes, 2 people have been cured of AIDS, or they can say that they have been sterilized of AIDS. I told you about a functional cure and a sterilizing cure. In the case of 2 people worldwide when we study, they say that these 2 people have been cured of AIDS completely and they have been in remission till then. We cannot say completely cured because they are still living and yet there is no evidence of the virus in their body. The first person is known as Timothy Brown. He is nicknamed as the Berlin patient. He was diagnosed with AIDS in the year 1995 and he also had leukemia. So his doctor, as a part of the research work, suggested that he undergo bone marrow transplantation, and a donor was to be selected for bone marrow transplantation. The doctor told him to try to look for a donor with mutation in the CCR5. You know the significance of CCR5, isn't it? It is an important co-receptor which helps in binding of the virus into the macrophage or any CD4 cell. So CCR5 is an important co-receptor which is involved in binding, and the doctor suggested to him to undergo or search for a donor with CCR5 mutation. That means the CCR5 should be negative or it should be not functional in that donor. Under such condition, if the bone marrow is transplanted, the Berlin patient who has AIDS will have CD4 cells free of the co-receptor, and then the AIDS virus will not be able to multiply in his system, because once you remove the bone marrow, replace it with a new one, transplantation. Once transplantation takes place, that person will be cured of AIDS. And so they got a donor surprisingly with the same CCR5 mutation, and later this person was checked for HIV and he was found to be negative. He was found to be HIV free. You can see the science behind it once more. CCR5 is a protein receptor which is used by the HIV as an entry point to the immune system. It's a very rare condition that a person has mutation for CCR5, and the name of this mutation is CCR5 delta 32 mutation, as a result of which the receptor will be small in size. So the HIV will not be able to bind to that receptor. So the donor which was selected in the case of Timothy Brown was a person who had CCR5 mutation. As a result, this person, when he was transplanted with a bone marrow having CCR5 32 mutation, he was found to be free from HIV, because HIV is not able to bind to this co-receptor nor infect the cells. And so he is the first patient who is said to be disease free, or you can say he is under remission from AIDS. And there is one more person who is known as a London patient. I leave it to you to find out who the London patient is and what happened to him and how did he become disease free. Now going to the list of some NGOs which are functioning in India to help spread awareness about AIDS and remove the social stigma regarding AIDS. Neptune Foundation, Snehalaya, Grace Peter Charitable Trust, Priyadarshini Seva Mandali, and finally the Swadhar Institute for Development of Women and Children. Now we have to understand some current research which is happening in the case of drug development in AIDS. As you are all children who might be interested in medical field, you will be also hearing a lot about medical research in the field of AIDS, and I would recommend it that you read extensively on this topic. So let us see what all has happened in the drug development in the case of AIDS up to date. I hope you remember this diagram. In this diagram you can see the viral entry and integration into the host cell. The genome enters, converts into DNA, enters into the host cell, and using the host cell machinery, RNA molecules are produced, and RNA and viral proteins are also produced simultaneously. They will be packed to form new viruses which will leave the cell to infect other cells. So this is a cyclic event which we have already dealt with. Now the medicines which are for AIDS are working on different steps of this process. Some medicines will cater to the binding of virus on the membrane of the CD4 cells. That means they will be blocking this particular step. They will be preventing either the binding to the receptors or in the process of membrane fusion. So there are classes of drugs which deal with this step. That means they will prevent the viral binding to the receptor on the surface of the cells or in the process of membrane docking and fusion. Then there are other classes of drugs which are known as reverse transcriptase inhibitors, which inhibit the reverse transcriptase enzyme. And most of the current drugs which people are using are functioning at this level. Then there is another stage in which the DNA is incorporated into the host DNA by using the enzyme integrase. This integration step is blocked by several other drugs. So that is another area where drug development is concentrated. Finally, the integrated provirus will start producing viral RNA. At this stage also there can be drugs which prevent the formation of viral RNA from the host DNA. Okay. Then so there can be drugs which inhibit at this level. There are also other drugs which inhibit proteins of the virus from being produced in the host cells. Inhibitors which prevent the proteins from being produced in the host cell. There are yet others which will prevent the replication of the virus by blocking the assembly of the molecules. So at all these levels, drug development is concentrated, and combinations of drugs are sometimes used, like triple drugs and all, which function at various levels of the process. As I told you earlier, you can also be contributors to drug development in AIDS. All these are still progressing, yet we have not got a complete novel drug which can actually block the viral entry and replication. I hope you have got sufficient knowledge regarding the topic AIDS. In the current session, we studied about symptoms, prevention, treatment, and some common questions along with the current research topics. I recommend that you read Medicine.net, then sites like Osmosis, journals like Lancet to know about AIDS more. It is a very interesting topic because still majority of human population is infected with AIDS, especially in Asian and African countries. Till we meet again, thank you and have a nice day.